of vaccines, right? So in the previous video we have discussed about a basic concept about vaccination, immunization and vaccines, right? So once you remember what are vaccines, it is very simple. Vaccines are killed or attenuated pathogens, or they are antigenic proteins of pathogens, right? So this is what we have discussed in the previous video. Today let us discuss types of vaccines. Okay, see, if you come to the types of vaccines, there are three types of vaccines. It is based on the upgradations, right? The first type of vaccine is called as first generation vaccine. We can write it as this is 1G. Okay, this is 1G. First generation vaccines. Second type of the vaccines are called second generation vaccines. This is 2G vaccines, right? Now the third type of vaccines are third generation vaccines. This is called 3G, right? It is very simple to remember. There are three types of vaccines. Number 1 is 1G, number 2 is 2G, number 3 is 3G. This is simply the upgradation of vaccines, right? First generation vaccine, second generation vaccines and third generation vaccines, right? You might have heard about your mobile phones, right? Upgradation takes place. Now we are using 4G, right? 5G also going to come, but in the vaccines you remember only 1G, 2G, 3G. So let us come to the first one. First generation vaccines. They are the first vaccines produced. Okay, first vaccines. As you know, that is first vaccines produced by traditional methods. This is produced by traditional methods, and the first vaccines are produced by using directly the microbes, right? Therefore, what are first generation vaccines? Now it is very easy to tell. So they are the vaccines developed by traditional methods by using microbes, directly by using the pathogens of that particular disease, and that pathogen only used for production of vaccines. That is what is the first generation vaccines. Developing vaccines by traditional method by using microbes is called first generation vaccines. In this first generation vaccines, again we have three groups. There are three groups under first generation vaccines. Let us see first one. The first one is killed vaccines. Killed vaccines. Yes. Now you listen. For example, you think that this is the virus. Okay. Think that this is the polio virus. Polio virus. This polio virus is causing polio. This is causing polio. Okay. Now, what scientist did is that scientist, his name was Sack. His name was Jonas Sack, one of the scientists, Jonas Sack. He took this polio virus and killed it, and that killed polio virus used as vaccines. Just simply killed the polio virus and used as a polio vaccines. Right, that vaccine is called as the SAC vaccines. Now, the very important thing is that while killing the pathogen, one care should be taken, because while killing the pathogen, there should not be an inactivation of proteins present in antigens. You know that is, this virus, on the surface of the virus, what are present? Antigens are present, and these antigens basically made up of proteins. So therefore, these proteins should not deactivate while killing. So that's why when I told about antigens, I told you that antigens have two properties, antigenicity and immunogenicity. So the first property, what is called antigenicity, should not be lost. Then only this can be used as the vaccines. Therefore, first one is killed vaccine. This consists of killed microbes. Right? One of the best example for killed vaccine is the SAC vaccine. SAC vaccine is nothing but it is the polio vaccine. It is the polio vaccine first discovered by the scientist Jonas Salk, and this vaccine was given as injection. It is given as injection. So this is what is I told in the previous video. Actually, there are two vaccines in the polio. The first vaccine was SAC vaccine, and that was given as injection. Okay. Now come to the second group under first generation vaccine. Second group, scientists thought they used live vaccines. The second group, that is live vaccines. So what are these live vaccines? Live vaccines means it consists of attenuated microbes. Attenuated microbes. Already I told you, what do you mean by attenuated? It is weakened microbes. Right? Now you listen. What Jonas Salk did, he took the polio virus and killed and used as vaccines. One more scientist came, his name is Albert Sabin. Albert Sabin. He took this polio virus and made it weak. He made it weak. He attenuated the polio virus and then he used as vaccines. Right? But here also one very important care is required while making weakening of this polio virus. This polio virus should not cause polio. The pathogenicity of the polio virus should be removed. Then only it can use as vaccines. So weak ಮಾಡಿಕೊಳ್ಳುವುದು, that is live vaccines. Right? ಅದೇ kill ಮಾಡಿಕೊಂಡ್ರೆ, that is killed vaccines. So therefore, the example for live vaccines is Sabin vaccines. Already I told this. This is also polio vaccine. It is prepared by scientist Sabin, Albert Sabin, and this vaccine is considered as OPV, oral polio virus, because this is giving by oral route. So ಅದರಿಂದ ಈಗ ಹೆಚ್ಚಿನ ಮಕ್ಕಳಿಗೆ ಕೊಡುವಂತಹ ಪೋಲಿಯೋ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಯಾವುದು? That is the Sabin vaccines. ಆ Sabin vaccine ಅಲ್ಲಿ ಏನಿರುತ್ತೆ? In that attenuated polio virus ಇರುತ್ತೆ. So that type of the vaccines are called live vaccines. ಮೊದಲು ಮಕ್ಕಳಿಗೆ ಕೊಡ್ತಿದ್ದಂತಹ ಪೋಲಿಯೋ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಯಾವುದು? ಸ್ಯಾಕ್ ವ್ಯಾಕ್ಸಿನ್ಸ್. ಆ ಸ್ಯಾಕ್ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಅಲ್ಲಿ ಏನಿತ್ತು? ಅದರಲ್ಲಿ ಕಿಲ್ಡ್ ಪೋಲಿಯೋ ವೈರಸ್ ಇತ್ತು. That is why that is an example for killed vaccines. Now come to the third group of first generation vaccines. If you see the third group, that is called toxoid vaccines. Toxoid vaccines. Yes. Now think that a disease called tetanus. As I told in the previous video, this is the disease caused by bacteria. The name of the bacteria is Clostridium tetani. This is Clostridium tetani. Right? Now, this bacteria, after entering into the body, this tetanus Clostridium produce toxin, right? This toxin is going to cause the disease called tetanus. You should know that is. So some of the microbes, they produce toxins. That toxins are responsible for disease. For example, you remember about the plasmodium. Plasmodium produces one toxin called hemozoin. That hemozoin is responsible for fever and chill. Similarly, tetanus produces a toxin that is going to, this bacteria produce toxin that is going to cause tetanus. Now scientists thought, so in order to give the vaccine to this disease, why we should give this complete bacteria? Now scientists thought that is, it is better to give the toxins. But if they give these toxin as it is in active form, definitely that toxin is going to cause tetanus. Then they thought that is, we should give inactivated toxins. If inactivated toxins of microbes is given as vaccines, that is called toxoids. So what are toxoid vaccines? It consists of detoxified vaccines. This consists of detoxified vaccines. Detoxified, inactivated toxins. Toxins na toxicity anna remove ಮಾಡಿ ಅದನ್ನು vaccines ಆಗಿ ಕೊಟ್ರೆ that is what is called toxoid vaccines. Here I have going to give two examples. One is tetanus vaccine and second one is diphtheria vaccine. Diphtheria, it is also a bacterial disease. The caused by bacteria, the name of the bacteria is called Corynebacterium diphtheria. So ಈ ಎರಡು ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ದೇಹದ ಒಳಗಡೆ ಬಂದ ಮೇಲೆ ಟಾಕ್ಸಿನ್ಸ್ ಅನ್ನು ರಿಲೀಸ್ ಮಾಡೋದ್ರಿಂದ so to this disease toxoid vaccines are given. Right? So there are three groups under first generation vaccines: killed vaccines, live vaccines and toxoid vaccine. Please remember this, and this is asked in NEET examination in match the following. Right? They will give examples and they ask what are present in that. Right? Once again you see, SAC vaccine contain killed microbes. Then Sabin vaccines contain attenuated microbes. Then tetanus vaccine and diphtheria vaccine contain detoxified toxins. Okay, this is about first generation vaccines. Now come to the second generation vaccines. Second generation vaccines means now this time what happened, lot of biotechnological technologies are implemented, lot of research happened in biotechnology. Now scientists able to produce the polypeptide chain of antigen. Right? Initially they were taking directly pathogens, or they were taking what? The antigens of the pathogens, or they are extracting the toxins of the pathogens. Now they are able to produce the protein, especially what? Polypeptide chain. So what What is polypeptide chain? It is nothing but protein. So producing the protein of antigens by using a technology called rDNA technology. So rDNA technology is nothing but recombinant DNA technology. So you just remember, this is the technology come in biotechnology. So you are going to study complete information about rDNA technology in the biotechnology chapter. But here you just remember, rDNA technology is the technology come under biotechnology. By using this technology, production of polypeptide chains of antigens. Antigens and proteins are produced modally. That's why this is the second generation vaccines. So second generation vaccines is that producing polypeptide chains of antigen by using rDNA technology. One of the example for second generation vaccine, this is very important for your competitive. So second generation vaccine, that is the hepatitis B vaccines. You know the disease called hepatitis, and hepatitis B vaccine is the second generation vaccine. This hepatitis B vaccine is produced by using the organism called yeast. Right? The organism used to produce hepatitis B vaccine, that is yeast. The technology used is rDNA technology. Remember, dear student, two questions can be asked here. One is, which is the technology used in the production of hepatitis B vaccines? That technology, the answer is rDNA technology. Second question, which is the organism used in the production of hepatitis B vaccine? Answer is yeast. Yeast is used in the production of this. Now come to the third generation vaccines. This is again upgradation. In the third generation vaccines, what scientists are doing or what the doctors are doing, that is, here doctors are injecting genetically modified DNA. Right? Genetically modified DNA into a body. Now they are not producing here any polypeptide chains. They are directly modified, means change. Ili DNA ಅನ್ನು change ಮಾಡಿ, DNA ಅನ್ನು change ಮಾಡಿ ಅಥವಾ genes ಅನ್ನು ಬದಲಾವಣೆ ಮಾಡಿ, that genetically modified DNA is injecting into the body. So after that what happens? Those genetically modified DNA produce one or two antigens proteins. ಇದು ಬಾಡಿಯ ಒಳಗಡೆ ಹೋದ್ಮೇಲೆ ಅಲ್ಲಿ ಆಂಟಿಜೆನ್ಸ್ ಪ್ರೋಟೀನ್ಸ್ ಅನ್ನು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡುತ್ತೆ. ಸೊ ಜೆನೆಟಿಕಲಿ ಮೋಡಿಫೈಡ್ DNA ಅನ್ನು ಇಂಜೆಕ್ಟ್ ಮಾಡೋದು, ಅದು ಬಾಡಿಯ ಒಳಗಡೆ ಹೋಗಿ ಅಲ್ಲಿ ಆಂಟಿಜೆನ್ಸ್ ಅನ್ನು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಿದ್ರೆ that is the third generations. So here also the biotechnological processes are involved. Right? Here producing protein and then that proteins are used as antigens. But here changing the DNA and that DNA is injecting into the body and that DNA produces one or two antigen proteins. The example for third generation vaccines are herpes vaccine and second example is influenza vaccines. Influenza vaccines. But dear student, the most important exam ಅಲ್ಲಿ ಕೇಳಿದ್ದು ಫಸ್ಟ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ ಮತ್ತು ಸೆಕೆಂಡ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ಸ್. ಥರ್ಡ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಬಹಳ ಕಡಿಮೆ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಇದುವರೆಗೆ ಕೇಳಿದ್ದಾರೆ, because it is not possible to do all the vaccines based on this concept. Right? Now, after learning these three type of vaccines, you might have heard about one vaccine called DPT. Right? You have heard that is called DPT. Now, the scientists thought in another way. Why we should give the one vaccines for one disease? One disease ಗೆ ಒಂದು vaccines, ಆಮೇಲೆ ಇನ್ನೊಂದು disease ಗೆ ಇನ್ನೊಂದು vaccines. ಹಿಂಗೆ ಕೊಡ್ತಾ ಹೋದ್ರೆ ಏನಾಗುತ್ತೆ, ಬಹಳಷ್ಟು ವ್ಯಾಕ್ಸಿನ್ಸ್ ಕೊಡಬೇಕಾಗುತ್ತೆ. ಆವಾಗ ಸೈಂಟಿಸ್ಟ್ ಯೋಚನೆ ಮಾಡಿದ್ರು, ದೇ ಕಂಬೈನ್ಡ್ 3 diseases vaccines. Here you can see this is one of the combined vaccine. In that vaccines are there for 3 diseases, that is called DPT. So what are the 3 diseases? Here D stands for diphtheria. This is diphtheria. This is the disease caused by the bacteria called Corynebacterium diphtheria. Here P stands for the disease called pertussis. Pertussis. Okay, this is commonly called as whooping cough. Right? This is commonly called as whooping cough. So in Kannada we say nai kemmu. So whooping cough. Then this T stands for tetanus. Tetanus. Andre DPT vaccines ಅಲ್ಲಿ tetanus, pertussis ಮತ್ತು diphtheria ಈ ಮೂರು ಡಿಸೀಸ್ ನ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಇರುತ್ತೆ. That's why it is called as combined vaccines. And remember, this is the first triple antigen vaccine. Triple antigen vaccine. Okay, triple antigens are present in this. This DPT vaccine is example for toxoid vaccines. Actually, it is the first generation vaccines. This is come under toxoid vaccines because DPT, right? Diphtheria, tetanus, in this detoxified toxins are used. So DPT vaccine is an example for toxoid vaccines of the first generation vaccines. Similarly, you come here, one more vaccines you might have heard about, that is called MMR, right? MMR. So this is also a triple antigen vaccines, but this is given to viral disease. Here M stands for the disease called measles, right? Disease stands for, that is measles, right? Another M stands for mumps, right? Manganabau, right? Sometimes you will get the mumps, Manganabau. This R stands for rubella. So these all are viral disease. ಈ ಮೂರು ಡಿಸೀಸ್ ಗೆ ಒಟ್ಟು ಸೇರಿಸಿ ಒಂದು ವ್ಯಾಕ್ಸಿನ್ಸ್ ಕೊಡಲಾಗುತ್ತೆ, that is called MMR. MMR ನಲ್ಲಿ ಯಾವೆಲ್ಲ ಡಿಸೀಸ್ ಗೆ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಐತಿ? measles, mumps, rubella. This is the example for live vaccines, right? This is an example for live vaccines. Therefore, you see here it is, I can add here one example. So for toxoid vaccines, that is DPT. I can add one more example for the live vaccine, that is MMR. MMR is an example for live vaccines. Now finally, you might have heard about one more vaccine, that is BCG. See here, BCG vaccines are the KCET. So this BCG vaccine, what's the full form of this BCG? BCG vaccine full form is Bacillus. C stands for Calmette, right? Bacillus Calmette guerin. Guerin. Bacillus Calmette guerin. So Calmette guerin is the name of the scientist he discovered or he prepared this BCG vaccine. This BCG vaccine given to the disease called TB. So TB ಅವರಿಗೆ BCG vaccine, TB ಬರದ ಹಾಗೆ BCG vaccine ಕೊಡಲಾಗುತ್ತೆ. TB is nothing but tuberculosis. It is a bacterial disease. The bacteria called the name of the bacteria is Mycobacterium tuberculosis. So TB ಗೆ ಯಾವ ವ್ಯಾಕ್ಸಿನ್ ಕೊಡಲಾಗುತ್ತೆ? BCG ಕೊಡಲಾಗುತ್ತೆ. ಇದು ನೆನಪಿಟ್ಕೊಳ್ಳಿ, ಇದು BCG ಅಂತ 3 ಲೆಟರ್ ಇದ್ರೆ this is not a triple antigens vaccines. This is only given to the TB. So this is about BCG, right? So this is about the types of vaccines. Here exam ಗೆ ನೀವು ನೆನಪಿಟ್ಕೊಳ್ಳಬೇಕಾದದ್ದು these first generation vaccines. ಇದು NEET ನಲ್ಲಿ match the following ಅಲ್ಲಿ ಕೇಳಿದ್ದಾರೆ, right? ಆಮೇಲೆ second generation vaccines ನ hepatitis B vaccines ಇದೆಲ್ಲ, ಇದನ್ನು they may ask in the theory for 1 marks. Right now 1 marks ಗೆ theory ಅಲ್ಲಿ hepatitis B vaccines ಕೇಳೋ ಸಾಧ್ಯತೆ ಇರುತ್ತೆ, because hepatitis B vaccines that is there in the NCERT book. So this is about vaccines. Once again we go through this. In the next video we are going to discuss about the human immune system. Till then, keep smiling. Thank you.